So, hello, ladies and gentlemen, or moin, as we say here in Hamburg. We hope you and your loved ones are all healthy and happy in these interesting times, and we welcome you today to our live broadcast on Optimize Your Bioprocess from shaker to bioreactor. This webinar today will be presented to you from Hamburg and Juelich, which is close to Cologne here in Germany, by my lovely colleagues Ines Hartmann and David Solbach. Both are amongst Eppendorf's most experienced specialists in regards to supporting scientists with their suspension cell culture applications, and I would like to give you a short introduction about both in a minute. My name is Christian Haberland. I'm Marketing Manager for Instruments and Systems here at Eppendorf, and I will be your moderator for today's webinar. At the end of the presentation, I would like to give you a short overview about solutions that might be helpful for you to improve your daily cell culture work. But before we begin, I would like to ask you for a favor. I would like to encourage you all to get as most as possible out of this webinar by submitting as many questions as you want at any time you want during the presentation. Just type them into the Ask a Question box on the left and click Send. We will answer as many questions as we have time for at the end of the webinar. And if you have any trouble to see or hear the presentation, just click on the Support tab at the top right or report your problem by just clicking on the Ask a Question box on the left. Continuing education credits can be obtained by simply clicking on the tab at the top right and just following the process. If you're interested in further material, we have prepared for you a little bit of download material that you can find under the Resources tab. And make sure you have your phone ready, because there are some of the QR codes which can simply be scanned via your telephone during the presentation. Now, let me quickly introduce both speakers of the presentation today. As I said, Ines and David are amongst our most experienced specialists that focus on suspension cell culture applications in shakers and bioreactors. Something about Ines. Ines Hartmann is application specialist focusing on cell handling and is responsible for the shakers and mixers here at Eppendorf since many years now. And she joined the company 12 years ago and covered many cell culture related topics since. She obtained her diploma degree in biology at the Humboldt University in Berlin, here in Germany. David Solbach is Scientific Communications Manager at Eppendorf, the Bioprocess Center. He studied plant biology and molecular genetics at the University of Uppsala in Sweden, and he investigated there the role of autophagy-related gene 9 in the regulation of autophagy in his master thesis. And he deepened his knowledge in working with yeast and bacteria for more than 3 years before he joined Eppendorf last year. So I'm happy that I could convince Ines and David to share their knowledge with us today, and I know and I can assure you that they have prepared a really nice presentation for you today, a really nice webinar. So therefore, I will shut up now and let's get started. Ines and David, the stage is yours.
Thank you, Christian. We are happy to give you in the next about 40-45 minutes some insights into bioprocessing. So we will have 3 parts for you. In the end, you will hear Christian again. In the introductory first part, you will hear what things should be considered prior setting up a bioprocess. In the second part, from shake flask to bioreactor, we go step by step through a typical workflow in setting up a bioprocess. So from the pre-culture via the selection of sensors and control up to different cultivation strategies. In the final section, we have a short glance on the process of upscaling. Results of bioprocessing are visible in our daily life. For instance, when we consume baked goods or dairy products or beverages like beer. These products are produced with the help of microorganisms. And food and feed is only one area of bioprocessing. Also, the huge pharmaceutical and life science industry uses cells as, let's say, microfactories for the production of therapeutic proteins, vaccines, antibodies, or new emerging cell therapies. Last but not least, important to mention, bioprocessing also helps in saving our environment. Sustainable technologies, for instance, the generation of biodegradable plastics and alternative fuels, are generated with the help of cells. With this, bioprocessing is an important tool in industrial biotechnology. So, as the title says, it helps to feed, heal, and fuel the world. So, what things have to be considered when setting up a bioprocess? The product is produced with help of microorganisms or cells, so they are the main actors in bioprocessing. Depending on the product, the first consideration should be the selection of the proper expression host and system. And here we have a first survey question for you, and I sent you the survey now. You should see it. So what expression host do you work with? I give you some minutes. Did I send? Yeah. Okay, I see. Send results to the audience. Yes, so you work with yeast. Now it's updating bacteria, mammalian cells. Yeah, it's a good mixture. So, yes, so the first thing we have to consider is the expression host, so the selection of the proper expression host and system. As example, we pick out here the typical expression host for therapeutic protein expression. So let's have a comparison about pros and cons. So in a bacterial expression systems, E. coli is the most prominent one. Bacterial expression systems are preferred. Genetic manipulation is easy. They achieve a high biomass in a short time. Plus, they are uncomplicated and cheap to cultivate. The downside is that they are prokaryotic. So in terms here of therapeutic recombinant protein production, they are not so well suited because they lack a system for post-translational modifications, which is required for proper folding of larger proteins. What about yeast? Yeast achieve high biomass in short time with a low-cost media, same like E. coli. As it is eukaryotic, it offers also some post-translational modifications. The drawback here is that yeast show different glycosylation pattern compared to mammalian systems, so they are not always suited. Insect cells are most similar to mammalian expression systems. Cultivation is relatively easy. Drawbacks here include that they grow relatively slow and the media costs are high, plus also the glycosylation differs from that of mammalian cell. So for the example, therapeutic recombinant protein production, mammalian cells are an ideal choice for the production. This system offers the desired post-translational modifications and proper folding. The main drawback here are only the costs of protein production. The cells grow slowly, the media is quite expensive, and to culture cells, mammalian cells is quite tedious, as the ones who work with mammalian cells probably know. And also they are sensitive to contamination. Nevertheless, you select now your expression host, and the next step would be the media selection. Media composition is a complex field, and we will not have the time to go in detail here. In short, it must include all nutrients necessary for higher cell growth and must also support product stability. Whereas microbial broths like LB or TB or YPD for yeast are often simple and self-composed, as you here see is like a powder very often, insect and mammalian cell media are more complex and mostly commercially available, like you see here. Media optimization and fine-tuning is done during process development as well. The next step is the selection of the proper cultivation system and strategy. Volume, volume is of course one selection criteria, so what amount or what yield of product do you would like to achieve. But second, we must choose the system our selected cells feel comfortable in. And for this, we have again a look at our organisms and their individual requirements. Whereas E. coli, as you see in the upper row, and mammalian cells prefer it warm at 37 degrees, yeast and insect cells grow better at slightly lower temperature. And also here we must look at the product too. A shift to a lower temperature is often required from the time of induction of expression of the protein. which is beneficial for proper folding and hence solubility of the protein, especially when you work with bacterial expression systems. Oxygen is the limiting factor for aerobic cell growth. So agitation, the second row, is one mean in shakers and bioreactors to enhance oxygen transfer to the culture. Mammalian cells and insect cells, as you may know, are more fragile than microbes. The challenge here is obtaining good mixing and delivering oxygen without damaging the cells. This is especially a problem at high density. Microbial cultures are not shear sensitive. High agitation speeds can be supplied. Here, due to higher metabolic activity and agitation speed, heat removal is the main issue, especially at larger volume, so cooling is absolutely important in the system. Last but not least, I would like to mention pH. pH optima also vary, whereas, for instance, let's take the baker yeast, Saccharomyces cerevisiae, grows better under slightly acidic conditions. Mammalian cells have a very tight optimum around a neutral pH. pH, of course, and the other parameters decrease and rise during process because of a metabolic process and must be kept stable in bioprocess systems. Now we come to the shakers and bioreactors, so they should provide the optimal conditions for growth. Agitation, both systems do, stirring or shaking, supports both nutrient and also oxygen supply, as I just said. In shakers, the oxygen supply is a passive process via the liquid swirling surface. In bioreactors, oxygen supply is a controlled process by a mixture of agitation and sparging. Sparging means active pumping air or pure oxygen to the culture. There are diverse other control options. Temperature stabilization, of course, both systems do by temperature control, shakers the chamber, and bioreactor by other measures you will hear later on. And bioreactors offer additional control beside DO control, so dissolved oxygen control, pH, and also feeding control. Both systems have their pros and cons. And if you are interested in getting more information about shaking and stirring, please visit our last webinar recording and scan the QR code. So, last but not least, after growing and expanding the culture and hopefully the product, the selection of the adequate downstream process is crucial to get the product from the culture in the needed quality, amount, and stability. This aims at purifying the product by separation, cell disruption, concentration, and many other techniques. But that is another big chapter which is out of scope for this webinar. And with this, we come to the next chapter. So you have decided for an expression host, selected the media, and have a first idea about what a bioreactor is able to provide. Now it is time to learn how to set up a bioreactor and learn what possibilities exist in monitoring and control and what kind of cultivation strategies you may choose. Here at that slide, you see a typical workflow for the setup of a bioprocess. We will go through it step by step. First, the inoculum for the bioreactor must be generated, often in a shake flask. Second, the bioreactor is set up for the process, so making connections, calibrate the sensors, and so on, until autoclaving if it's not single use. And once the bioreactor is prepared, medium can be inoculated. With starting the process, the next step is monitoring and control, according to the cultivation method you choose. And the last step is harvesting for further downstream processing. So we start with the bioreactor inoculum. The first step is the preparation of the bioreactor inoculum. That means producing a certain amount of biomass, which is introduced to the broth media of the reactor to start the process. Mostly, these precultures are grown in shake flasks. Sometimes also smaller bioreactors serve to grow precultures, for instance, for large production scale fermenter volumes if the preculture itself is 30 liter. And the main objective is to achieve a short lag phase in the main culture so that a quick process start can occur. Second aim is a reproducible growth from batch to batch. So, there are certain things to consider. An optimal inoculum must be available in the vital state, as you remember the aim of keeping the lag phase short. It should be harvested in the logarithmic phase of growth. With the cells in a vital state, it gives a good chance of fast adaptation to the reactor environment and a short lag phase. Of course, the inoculum must be available in a sufficiently large volume. This depends on the size of the reactor, what volume is required. It is very important throughout all the preparation and especially also at the introduction to the bioreactor that you apply aseptic techniques, that there is no transfer of a contaminant into the main culture. This can be cost intensive, especially when we talk about mammalian culture. In the end, the pre-culture should be consistent in quality, establishing an efficient and reproducible process. So how to prepare inoculum? Inoculum is prepared from the preservation culture, meaning coming from -80 or liquid nitrogen in terms of mammalian cells, and activates the cells prior to their usage for fermentation. So let's have a look at microbiology first. It is beneficial always to use a single colony from a freshly streaked agar plate to inoculate a liquid bacterial culture and not go directly from glycerol stock to liquid culture. That not only prevents a potential enrichment of contamination if having one in stock, hopefully not, it also results in a better synchronization of strains if your aim is to run comparable experiments, for instance, screenings. So from the small liquid culture, volumes can be further expanded to larger flasks depending on the required inoculum volume needed. This is called then seed train. So long seed trains are required in industrial settings where the inoculum of, say, a 300-liter reactor needs to be itself a rather large volume, 15 to 30 liter, for instance. For cell culture, the vial is thawed according to cell-specific SOP, that is depending on the cell type, cell line, in a small volume and either static or shaken and expanded by splitting cells. And here the expansion process is called seed train as well. It is important to not inoculate at too low volumes because lonely cells don't grow very well. So typical volumes for microbiology, NOD of the pre-culture between 2 to 4 or 1 or 4 is common, and depending on the media, of course, and the inoculum start OD in the main culture is usually between 0.3 to 0.5 in a volume of 2 to 5% of starting volume. As said, the optimal OD value depends on the culture and the medium. In cell culture, a density of 500,000 cells to 1 million cells per milliliter in a volume of 10 to 25% of the starting volume of the reactor is common. So, let us remember some slides ago, one aim was a constant quality. So, how to standardize inoculum quality? So, once established an optimal OD, it is good to reproduce this for the next batch. The normal procedure you see here is growing an overnight culture. Here the harvesting time varies or requires a very good planning from your side, staying late or coming very early to the lab. So the risk that the culture is not in the target OD is high, and using an inoculum that has entered the stationary or even worse in the death phase may lead to a prolonged lag phase in the bioreactor. You have many dead cells in your inoculum. A programmable shaker with the possibility to pre-program temperature and RPM in the chamber helps to schedule better your inoculum preparation. If you know the time frame that is used to reach the target OD, you can pre-program a defined time the shaking is cooling. Here, for instance, 10 degrees, slow RPM, that the cells are a bit adapting to the media. And then in the second step, you add a step of the growth temperature, 37, and the cells grow. And then you come in the lab and the OD is in the log phase, and the cells in your bioreactor are happy. And last but not least, you can make it even more convenient than just control the chamber conditions. It is even more convenient to monitor growth in shake flask, for instance, in real time, and get a notification at the target OD. And the cherry on top would be pre-programmable seeding, and I would show you here an example. We integrated, so we have the shaker, the pre-programmable shaker, that is from us in this case, of course, and we integrated the CGQ online monitoring system from Aquila and the LIS automatic injection system, also from Aquila, and monitored a yeast culture. And as you see here, we programmed first a preconditioning 10 degrees with slow shaking, adaptation of the culture, and you can expand this phase as long as you want, depending on what time you would like to be in the lab. Then you have the seed inoculation with the LIS system, and also a ramp up of the shaker to 37 degrees and higher RPM growth monitoring, and you get a note when you have your target OD. So if you are interested in, please scan the QR code to have the application note. Okay, that was enough about the inoculum. It's time to go into the bioreactor, but before we start, I hand over to David. I have one more question for you, and I would like to know what type of bioreactor or fermenter you use. And I send the survey now and give you some time. Okay. I see most of you, as you see also hopefully as well, work with benchtop reactors, and also many don't work with bioreactors yet. And some work with, large production. Yeah, good mixture, but benchtop is the winner, I think. Okay, and now the pre-culture is ready for inoculation, but I think, David, we have to set up the bioreactor first, so I keep it cooled a bit more.
Exactly. It takes a little bit more before we can start with the real bioprocess process with the bioreactor. Thank you, Ines. And as I just see, there's also a huge part of you who didn't start working with bioreactors yet, so maybe I can convince you that you should consider going the next step. And if you're afraid to do so, maybe you're not afraid anymore after this webinar. As you did see, there were already a lot of aspects to consider for the first step of the bioprocess workflow. And I will now walk you through the next steps, and starting with the setup and the inoculation of the bioreactor, and then talking a little bit more about monitoring and control possibilities. Before the pre-culture can be transferred into a bioreactor, there are a few steps that need to be prepared. It's recommendable that you start these preparations in parallel to the pre-culture to optimize your workflow and save some time. If you want to have a detailed description of the entire process, this can be found in our short protocol 32. Just use your standard camera app on your mobile phone and scan the QR code to download the application note. A bioreactor is more than just a vessel. It consists of many different parts that need to be connected, sterilized, and equilibrated properly. What you can see here is a bioreactor that is ready equipped to be autoclaved. The headplate of a bioreactor contains industry standard PG 13.5 ports that enable the easy installation of sensors, cooling fingers, feeding and harvesting lines. And depending on the experiment you want to do, each bioreactor setup can be customized. I will now walk you step by step through the different parts of a bioreactor and explain for what they are good for. And let's start with the most obvious one, the motor. The motor is essential to control the steering speed, of course, of the impeller. And here already comes the first important parameter that can be precisely controlled and has a huge impact on the culture behavior: the agitation. The agitation is always important to monitor and control because, as Ines just told you, microbial cells are not that sensitive to shear stress, but on the other hand, you also have cell culture applications or even stem cell applications that can be performed in bioreactors, and these are really sensitive to shear stress. So you need to find a good balance between a homogeneous suspension and shear stress. The faster you steer, the higher is the stress for your cells, but the more efficient is the oxygen transfer from this gas phase to the liquid. What you can see here are a couple of different impeller types that can be used in bioreactors. And with each impeller type, the flow direction can be different. Radial flow, for example, occurs when the fluid is pushed away from the impeller's axis toward the vessel's wall. On the other hand, you have an axial flow, which occurs when the fluid is pushed up or down along the axis or the shaft of the impeller. The orientation of an impeller, if it's left or right-handed, and its agitation direction determine whether the direction of the axial flow is up or down. So always keep an eye on the impeller type and the agitation direction. A Russian impeller causes radial flow, whereas a pitch blade impeller causes a combination of both radial and axial flow. And if you're working with really stress-sensitive cells, you can also consider to work with a marine blade impeller. The leading phase of these kind of blades can be flat or concave, whereas their back sides are convex. This produces also an axial flow in addition to the radial flow. The most commonly used impeller types are the Russian and the pitch blade impeller. The Russian type impeller is used mainly for fermentation processes, since it ensures really good oxygenation of the solution but causes high shear stress to the cells. On the other side, you have the pitch blade impeller, which is commonly used for cell culture applications at lower RPM. And as I just said, if you have really sensitive cells, you can also consider working with marine blade impellers. And there are even more choices that you can think of working with. For example, there are special impeller types such as packed bed impeller or spin filters. These impeller hold the cells, for example, inside a cage attached to a growth matrix and protect them therefore from shear stress. Another advantage here is that they can be used for perfusion processes, whereas medium is extracted from the bioreactor without the withdrawal of the cells. And now don't be shocked, this is a typical German slide with a huge table and a lot of information. But don't worry, you really don't need to read it. It's just a guidance if you feel unsecure which impeller is the best for your application. So we have this summed up in our application of 315. If you are interested in this table, feel unsecure, just drop us a note or contact our specialist. We are happy to help you out here. The next component of a bioreactor is the aeration. To ensure that your cells always have enough oxygen, or in case of anaerobic fermentation, have no oxygen in the media, gas is added either to the headspace or directly into the medium with a submerged spatula. The spatula can have different shapes and pore sizes to adjust the amount and the size of the bubbles, since also the bubbles can cause shear stress to the cells. And another important accessory, of course, are the sensors that can be inserted to the bioreactor. On the right side, you can see a standard empty head plate of a bioreactor, which has quite a lot of empty holes. So if your experiment requires a lot of measuring and monitoring, there's enough space to add your sensors you want to choose. And if you don't need so many, you can just close the holes and use less. The information of the sensors, of course, should be used, for example, to control the temperature within the vessel. Temperature control is very important to ensure that your cells are happy, grow nicely, and of course produce a lot of your target product. Yeast cells, for example, tend to throw out the plasmid that you transformed into them if the temperature is too high. On the other hand, the growth rate drops significantly if the lower the temperature is, as you did see before with the example of the control temperature in the shaker. However, in the case of an E. coli fermentation, it is really recommendable that you have several cooling mechanisms available. 1 liter of an E. coli culture produces around 150 watts of energy. If you remember the heat of an old 100-watt light bulb, you can imagine that this is that this heat would be really dangerous to your culture if it wouldn't be controlled, and will also harm your cells or maybe ultimately destroy your bioreactor. So now you have an overview of the different parts of the bioreactor, and it's time to inoculate the vessel. The inoculation itself can be done fairly easy with a syringe through the feeding ports, or in case you're working with larger volumes, you can also automate this process and use the built-in pumps of the bioreactor to feed the larger bioreactor. In case of an experiment with E. coli, we recommend a final OD600 of 0.3 to 0.4 in the starting volume of the bioreactor. At the same time, the inoculation volume should not be more than 2 to 5% of the starting volume in your bioreactor. I also told you a lot why the bioreactor is more than just a vessel, but on the other hand, it is just a vessel. So a bioreactor alone cannot run a bioprocess. It needs a control system that controls and monitors all the process parameters. For this, the base unit has a couple of connections, for example, for the motor, the sensors, gassing, and the pumps. Additionally, modern bioreactor control systems come with a user-friendly and sophisticated control software already on board. Which brings me to the next chapter, monitoring and control of your process. As you can see, there are a lot of steps that can be controlled and a lot of biological parameters that need control. As I just mentioned, the temperature, the pH, and the dissolved oxygen concentration are one of the most important ones, but also mechanical parameters such as foaming and agitation should be controlled. And in addition, the big advantage of bioreactors is that you precisely can control the different feeding strategies, like as a batch or fed batch or even a perfusion process. But I will come to these different feeding strategies later in this talk. If you know your process, you can control your process. And even further, if you really know your process, you can predict the outcome. This concept is, for example, the core of the quality by design aspect. With the help of software, you can precisely monitor important process parameter and react accordingly. Software-guided monitoring also enables you to run multiple bioreactors in parallel without losing control. The software can even do more for you. It can take care basically of everything. I will show you some examples here, how the software controls the process. In this case, I will show you a pH control loop with our new Cybario 2 and bioprocess control system. In this setting, the control system was programmed to automatically maintain the pH at a value of 7, indicated by the dark blue line at the 7.0 value. In the course of the experiment, the metabolic products of the cells lead to an acidification of the medium, resulting in unfavored conditions for the cells if it's not controlled. But as you can see here, the system constantly monitors the pH and automatically activates or deactivates the acid or base pump to maintain the pH in a really narrow range around 7. In this case, with just a deviation of around 0.01 units. Another possibility how you can use the software and in combination with the sensors is the implementation of a feedback control loop via direct OPC UA connection. In this example, researchers from Resolution Spectra integrated their Raman analyzer to program a feedback control loop for glucose feeding, based on the real-time measured glucose concentration in the bioreactor itself. The analyzer communicates directly with the biocontroller and sends the glucose concentration that was read in real time in the bioreactor to the software, and the software then controls the feeding pump. By this, what you can see here, you can see on the left side a classical fed batch run, where you can see that the glucose concentration drops over time and is feeded batch-wise to have always a higher glucose concentration, around 3 to 5 grams per liter. On the right side, you see the feedback control loop, where it was programmed in a way that the glucose concentration, once it reached the concentration of 5 grams per liter, the automation kicks in, and the concentration in the bioreactor of glucose is kept at 5 grams per liter. This is the blue line in the middle, which first drops and then goes into a linear phase. Here you can see that the Raman analyzer really precisely detects the glucose concentration, and via the feedback loop, the bioprocess controller maintained this concentration at exactly 5 grams. And at the end, the feeding goes down. This has a simple reason: the maximum working volume of the bioreactor was reached. The software recognizes this, and of course you don't want that your solution runs out of the bioreactor. The system also knows this and stops the feeding, and the process ends. The effect here, or the positive effect of this automated feeding, is that the overall lactate concentration, so the metabolic product that you always have in a process, but the lower the better. You can see in the feedback control loop, the lactate concentration is lower compared to the fed batch run. Another important parameter that needs to be controlled is, of course, oxygen, as we mentioned already. In a classical E. coli fermentation process, oxygen is generally the limiting factor. As you can see here, the amount of oxygen that is needed from an aerobic microbial organism is up to 20 times higher compared to the cell culture, compared to that of a cell culture run, which is also one point that explains why E. coli produce such a huge heat, or such huge amount of energy when they grow. A typical way to control and to ensure a sufficient oxygen supply to the culture is displayed in this 3-step DO control cascade. You could theoretically start directly from the beginning and gassing the bioreactor with pure oxygen, but this is expensive and we all want to save money in our daily lives, so we don't want to do this. But there are possibilities that the bioprocess controller controls this in this 3-step cascade. The easiest way in a fermentation process is that you start increasing the agitation step by step, the more oxygen you need. The higher the agitation, the better is the oxygen transfer to the medium. But at one point, the agitation reaches its maximum, because either it's more like you have a mixer that starts destroying your cells, or the foaming issue will be too much, or there are other reasons. So the next step would be to increase the gas flow to the medium. To save again a bit of money, you don't start directly with oxygen, you use standard air, where you have the our standard amount of 20% of oxygen. So with increased airflow, you also have then increased oxygen transfer to the medium. At one point, the E. coli had reached such a huge density that this is also not enough, and then is a point where the pure oxygen kicks in, and you have really good oxygen supply also at the end of the process. In a cell culture run, one is limited in increasing the agitation rate or in gassing, because both increase the shear stress to the cells. So there, the optimization of the DO control is a little bit more tricky, but on the other hand, as I just showed, they also don't need that much oxygen. Real-time off-gas is an important tool for gaining insights into a fermentation process by allowing the user to examine carbon evolution, oxygen utilization, and the transfer during the fermentation. This information can be used to find changes in metabolism, spot potential problems, and trigger changes in the fermentation process. Through off-gas analysis, insufficient oxygen or substrate supply can be identified. The graph on the right-hand side shows the oxygen transfer rate, the carbon transfer rate, and the respiratory quotient of a fermentation process. I won't explain these values more in detail in this webinar. You can just trust me that it's true what I'm telling you, because otherwise the slide would take too much time. But the signal, as you can see, drops significantly at the end of the process. And this signal drop indicates the end of the process itself, when the main carbohydrate source was depleted. This change in signal could be used to start the feeding for a fed-batch culture, for example. So you can even use the oxygen concentration, which is red, and the off-gas to control your glucose feeding pump, for example. And I mentioned it already, but especially in fermentation processes, the increased agitation and the gassing rate causes foaming problems. Foaming can be monitored with the help of level sensors that can distinguish between foam and liquid. Once foam is detected by the sensor, a pump can start that adds the anti-foaming reagent. This prevents foam out events and is especially useful when you are running longer experiments overnight or over the weekend. And now I did explain a lot about the different parameters that can be controlled and monitored by the system. But I didn't talk about the most obvious one yet, the feeding strategies. I will briefly describe you the major feeding strategies that can be applied in bioreactors, starting with the batch run, followed by the fed batch feeding strategy and the continuous feeding strategy with an example of an E. coli fermentation. At the end, I will also shortly show you a perfusion run performed with cell cultures. First, we have the classical batch fermentation. This fermentation process you can also call a controlled shake flask process in a bioreactor. You have the bioreactor and the initial medium is transferred into the vessel to a certain volume, for example, the maximum working volume of a bioreactor. And then the process is started and controlled by the bioreactor controller. The process runs as long as the carbon source is available, and once the carbon source is depleted, the cells don't grow anymore and you can stop the run and harvest the cells. As you can see here, the maximum OD in a batch run in this case in a bioreactor was around 90. The next step to improve this process would be a fed batch run, which I mentioned with the Raman analyzer example, for example, before. In a fed batch run, the initial volume in the bioreactor is lower, but once the process starts and the carbon source is depleted, the additional carbon sources are added step by step until the maximum working volume of the bioreactor is reached. With this, the OD 600 that you can see here reaches a way higher value. In this case, a value of 250, so nearly 3 times higher than a classical batch run. To even further increase the yield, not the OD in the bioreactor, but the overall yield, you can do a continuous feeding strategy. In a continuous setup, the initial medium is transferred to a bioreactor and inoculated, similar to a batch process. And you can also, for example, directly start with the maximum working volume of your bioreactor. But after the initial carbon source is depleted, you have at least 2 pumps that are started in parallel, and one pump starts feeding fresh media into the bioreactor, while the second pump starts at least the same rate harvesting of the media. With this, new carbon source is added to the controller, while the old medium with the cells is harvested. Due to the continuous harvesting of the cells with the medium, the maximum OD is, as I said already, it's lower, but the overall yield can be higher, so this process run can last for quite a long time. To compare these different fermentation modes, you can download our application note where this is described in more detail, which is the application note 408. And as I already said, last but not least, for the process strategies, we have the perfusion mode, which is very similar to the continuous manufacturing process, with the difference that you have a cell retention device connected to the harvesting tube. So once the carbon source is depleted, you start harvesting the media, but not the cells. So for example, in cell culture runs, you can cultivate your cells on microcarriers, and the microcarriers remain in the bioreactor while the medium is harvested. With this, the old medium with the cell metabolic products are harvested, and also with the product you want to have is harvested, and your cells grow to a higher cell density. Special impellers for this are, for example, spin filter or packed bed vessels, where the cells, which maintain the cells in the bioreactor. And with this, I hand over to Ines again.
Thank you, David. That was a lot of useful information. So with this, the upstream process is finished, and after every up comes a down. So here, not in a negative sense, of course. So no matter what process chosen, in the end stands the harvesting. And the separation of a protein of interest from a reactor is the first step in a downstream process. The selected harvesting method must be carefully chosen and would give enough food for another webinar content. So shortly, it depends on the type of cells, product, and properties of the process fluids. So traditional techniques include, for instance, centrifugation and diverse filtration techniques, disc stack filtration, tangential flow, and so on. So with this, we are nearly finished. In the last chapter, we will just have a short glance, so we go up again, on what it means when a process is upscaled, and have a short look at some scale-up criteria. So many products of bioprocesses, as you saw on the very initial slide, are of industrial use and must be produced in large amounts. Just think about vaccines or biopharmaceuticals. So scaling a process means a stepwise transfer of an established process in small volume to a large volume, with keeping the same productivity and quality. The stepwise transition is needed as starting in production scale is usually not cost effective, of course. So a typical scale-up workflow, how does it look? In the beginning of a typical scale-up workflow stands screening and media design, so the search of a productive strain and testing of optimal medium compositions. These early stages are often performed in shake flasks or even plate formats, as they allow a high amount of experiments to run in parallel in low volume, and that saves cost. For further screening and optimization, a parallel bioreactor, a mini bioreactor, provide individual control of up to 24 vessels, in this case, in parallel. So if, for example, just that you have an idea, you would like to compare protein expression at different temperatures, this would be the ideal choice, for instance. So next up starts process development and technology transfer. Technology transfer to pilot or production scale systems. So bench scale systems provide a great tool for this. The working volume, and then comes the pilot and production in a very large bioreactor. So the working volume in such a scale-up workflow can range from like 250 ml scale when you start from a bioreactor, or below 1 milliliter if you start with deep well plates, for instance, up to 2,000 liter working volume in production scale. And that is challenging. And for this, there are different scale-up strategies developed. So the general strategy is to keep one process parameter constant throughout the scale-up process. That could be either the impeller tip speed, that does not mean that means the end of the impeller, so the tip. It can be also the mixing time. Another very often used parameter is the KLA, so the volumetric mass transfer coefficient, that gives is a measurement of the capacity of the bioreactor to transfer oxygen into the culture under a given setup and under given conditions. And the gold standard at the moment is the impeller power consumption per liquid volume, power over volume. And as you see here, it's getting quite complex, and to explain each strategy would fill another webinar, and we are already at the end of our webinar. And this is exactly what we decided. And if you want to get more insights into scale up and explanations of what I just started on the last slide, join our upcoming webinar focusing on cell culture scale up. And it's at January 14 in 2021, also via the LabRoots site. And with this, I will hand over to Christian again, who will show you some of the Eppendorf solutions for cell cultivation and bioprocess. And after that, stay tuned. We will have some more time for your questions.
So, thank you very much, Ines and David, for this great presentation. Before we will start with the last poll question and the Q&A session that you've all been waiting for, I promise to give you a quick insight into the helpful solutions for your daily work. So let's go right to them. You probably all know that Eppendorf offers a wide variety of CO2 incubated shakers, incubated shakers in general, CO2 incubators, and also different bioreactors for different stages in development and also in production. And I would like to highlight here 4 products in this case. And the first one is the incubator shaker Innova S44i, which is perfect for reproducible inoculum preparation because it offers tight specifications regarding temperature and CO2 control, and also offers low vibration because of the construction of the drive that you can see here on the right side. It also offers reliable bioprocess planning because it comes with multi-step programming, and it is also compatible with automated feeding and biomass monitoring systems. And also for your audit safety, it comes with comprehensive documentation. So it logs alarms, chamber temperature, RPM, door openings, and also via user management, you can restrict the access to change any settings. If you want more information, just go to eppendorf.com/S44i. The next device I would like to show you is a special one. It's called the New Brunswick S41i, which also supports reproducible inoculum preparation with effective protection from medium evaporation by a relatively high humidity and also tight CO2 specifications. It's unique because it offers a static shell for incubation of adherent cells, but also a shaker platform, so you can do both cultivations under the same conditions that you can see here. And it's also quite special in the shaker world because you can easily and safely set it up for the next project by an integrated 120-degree disinfection. There's no fan inside and has smooth chambers, so you can quickly clean it inside, and you're ready for the next project. And also it provides a lot of high contamination prevention or protection. And what you can also see in the graph here, it's also very nice that you can save a lot of CO2 with this, and it also saves time because you don't have to change the gas cylinders that often. If you're interested, eppendorf.com/S41i. Now let's come to the bioreactors. The first one is the BioFlo 120 that goes under the topic performance meets value. So with one-touch processing for microbial or cell culture processes that it offers, you can see on the right-hand side here. It gives you a graphical view of the process data for the user. And also you can control a lot of user-defined loops and cascades. So it's perfect for automated control of critical process parameters. It gives you a lot of flexibility for your processes. And if you're interested, go to eppendorf.com/bioflow120 or just scan the QR code here. And the last one I would like to show you, we're pretty proud of this new device. It's called the Cyvario Twin, which we have been launched just a couple of weeks ago. It's the next generation bioreactor control system. So there are 19 different vessel variants that are available, glass or single-use vessels in case of the BioBlue vessels. And that's perfect for stem cell culture, and fermentation applications. It offers you a lot of flexibility and advanced application efficiency. If you want to know more about it, go to eppendorf.com/Cyvario. So with this, I would like to come to our last poll question so we can keep improving our webinars for you. And my question is, how do you rate the content of today's webinar? I will send it out just now. And with this, I would like to go directly over to the Q&A session. It's time for questions. So if you have any questions you would like to ask David or Ines, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many questions as we have time for. So let's get started. I already see that there are a lot of questions in here, and I would like to choose the first one. The question is, do you recommend working with baffled flasks or non-baffled flasks? I think, Ines, you're a perfect person to ask this question.
Yeah, I think this is related to a shaker and shake flask. So, yeah, that definitely depends on the type of cells you work with. So baffled flask, maybe not all know what it is, have defined cavities on the bottom, or with new designs also a bit far up. So, and they generate some kind of waves, and with this they improve like oxygen transfer to the culture. So indeed it could be helpful if cultures oxygen requirements are very high, or if you only apply low shaking speeds. Also for viscous cultures it's actually good. So if someone works with fungi, for instance, to prevent spore aggregation or culture pelleting. But on the other hand, you have also a higher foam formation, of course, because of this wave and turbulent swirl. So that may, if this goes up to the top, to the plug, it may hinder the oxygen transfer, but there may be solutions to use anti-foam as well. So, yeah, concerning inoculum, if that was your question, there is literature saying as well that baffle flask gives considerably more variable results because of the more chaotic flow behavior. Let's say that is with glass, it's a bit, with molded plastic, the variance between flask are not that high. With glass, there could be also variances between the vessels, so reproducibility could be one downside. But I think that is just a try with your culture and could be a solution or not, but definitely improves oxygen transfer to the culture.
Right, Ines, thank you very much. I have another interesting question I've already heard a couple of times during this webinars, and this one probably goes to David. And the question is, which impeller is suitable for stem cells?
Yeah, thank you for the question. So as I mentioned during the different impeller types, the Russian type is not the impeller you should use because this just causes too high shear stress. I recommend to use the pitched blade impeller because this is generally used for cell culture applications that causes low shear stress and good distribution of the cells within the bioreactor. Or what you can also do is we recently launched a special developed 8-blade impeller. This impeller was really developed for culture that are cultivated on microcarriers or in solution on aggregates. So, and since you also quite often cultivate stem cells as aggregates in your bioreactor, this impeller is also recommendable because it keeps your cells as in solution really at really low RPM speed. Yep, that's from my side.
Yeah, thank you, David. Here's another interesting question. So the question is, do you have any recommendation for pipetting high viscous glycerol? Who wants to take on this question?
Oh, okay. That is not shaker and bioreactor related, but probably it's about media preparation. If you pipette glycerol for bacterial media, terrific broth or something. Yes, so one solution could be to use like positive displacement pipette. So usually, maybe you all know you have an air cushion pipette and you cut the pipette tips, so that is not a good strategy for reproducible transfer of liquids. So if you, it's called stepper or multi-pet and has tips which don't have an air cushion in between. And there are also special tips for viscose liquids from us, they call visco tips. You can use for these high viscose liquids. So no air cushion pipette, but a stepper or a multi-pet. I hope that is answered with this.
Thank you, Ines. I think that answers the question fully. Here comes another bioprocess-related question, and the question is, how long does the sensor calibration take for setting up a bioreactor? Does this have to be repeated for every batch setup, David?
Give me a second, because this question is not answered that easily, because this really depends on the sensor type you're working with. Let me start with the DO sensor. For example, if you're working with an ISM or an analog sensor, you need to do at least 4 hours of polarization prior to the calibration. So you have already here 4 hours of waiting time, which is perfect for the inoculation preparation. Also, these sensors, you have the electrolyte, which I recommend to exchange prior to every one. Theoretically, you just need to do this after every 3rd autoclavation cycle, but to be on the safe side that you don't forget this, I just recommend to exchange the electrolyte prior to each run. So this is with the analog or ISM sensors. When you have an optical DO sensor, this is quite easy to use. Since it's optical, you just plug it in and you can calibrate it. This goes faster, and the calibration takes around 1 hour. So you have either this 1 hour calibration time or the 4 hour calibration time. When it comes to pH sensors, it again depends on the sensor type. If you have a gel-filled glass probe, that needs to be connected to the controller and calibrated before you insert it into the vessel and autoclave it. So again, here I recommend to take some time. So spending more time on the calibration is always better, because then the overall monitoring in the actual run later is way more precise. Especially with the gel-filled pH probes, I always recommend to insert So normally you use the pH buffer of 7 for the first buffer calibration point, and I would wait at least 10 minutes or even longer to wait that the gel is hydrated enough and that there are no fluctuations during the calibration procedure. The calibration itself of a pH sensor is faster compared to a DO sensor that takes, depending on your reading system, around 10 to 15 minutes, I would say.
Yeah, thank you, David. The next question I've chosen is a question that's related to antifoam. And the question is, you said antifoam is mandatory in fermenters when working with bacterial fermentation and high speeds. Is antifoam also used in shake flasks, Ines?
Yes, I think I mentioned it in the first question already. Of course, you can also use antifoam in shake flask, especially when you work with baffles, that is helpful. And if you work, for instance, with very high speeds paired with high fill volumes. So, for instance, ultra-yield flask have a higher filling and recommend to go to high speeds. So here it is definitely recommended and does not harm the cells.
Thank you. So last question that I've chosen. So there are a lot of questions actually in our chat here, so I have to choose wisely, and I choose one that covers both shakers and bioreactors. And the question is, what fill volumes do you recommend for bioreactors and shakers? Who wants to start?
Ladies first.
Okay, for shake flasks, it's general. The general rule is not more than 20%. It's one exception again in these. There are new designs developed. I mentioned these ultra yield flasks. I think there are more. They have specific design and baffle design, so they allow higher volumes. Plus, also this is for microbial cultivation. For cell culture, the fill volumes can also go higher. 40% as the oxygen demand is not that high. And the bioreactors?
The short answer would be it doesn't really matter. Read the manual. But we have recommended minimum working volumes and, of course, a maximum working volume. Taking our 3-liter BioBlue as an example, in theory, I think around 3.7 liters of medium fit into the bioreactor, but the maximum working volume in this case would be 3 liters. And vice versa, I always recommend to work with at least with the minimum recommended working volume, which we also tell you for each bioreactor. This ensures that when you start steering, that the sensor tips are always in contact with the medium, for example, and that the impeller is also covered by the medium.
Thank you both. Thank you also for your time here, for your great presentation. Do you have any final comments for your audience?
Thank you all for listening. It's a pity that we don't see you. It's still for me a bit strange. It's we did a couple of webinars already, but it works better each time. So for me, it was a great pleasure. Hope to see you again.
Also from my side, thank you very much for listening, and I hope we could answer all your questions. If they are, I see there are a lot of unanswered questions. We will come back to you via mail, so don't be afraid you are not forgotten. And if you have further questions, don't feel afraid and send us a mail, for example, to bioprocess-experts@eppendorf.com. Thank you, Christian.
Thank you very much. Yes, before we go, I would like to thank you all for joining us today and for your interesting questions also. As David said, the questions that we did not have time for answering today and those that you have submitted during the on-demand period will be addressed by the speakers and the contact information that or by the contact information you provided during the registration. I would like to thank Ines and David again for their time today and for their brilliant presentation. I would also like to thank Michelle Ashton and the whole LabRoots team for supporting us today. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you all to share that email with your colleagues who might have missed this event today. So we say goodbye from Hamburg and Juelich. Have a nice day, and most importantly, stay happy, healthy, and help to protect others. Thank you very much, and goodbye.